Have you heard what's happening in Calgary, Canada? Home to some of the world's best researchers and innovators in life sciences, Calgary is advancing healthcare solutions to solve global challenges. Calgary's dedication to the life sciences sector is evident in its labs, hospitals, schools, and the minds of its people, with its top institutions producing internationally recognized research and more than 110 life science companies backed by a highly skilled pool of talent. The life sciences sector is accelerating innovation in Calgary. If you're a bright mind or a bright company, Calgary is just the place for you. Take a closer look at calgarylifesciences.com. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast, and the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. The discovery and cell line development of biotherapeutics has been traditionally a distinct process. AbSci is taking what it describes as a more holistic approach by collapsing the process down and addressing the functionality and manufacturing of therapeutic candidates simultaneously. We spoke to Sean McClain, founder and CEO of AbSci, about how it is using AI and synthetic biology to reinvent the discovery process, how this is expanding the therapeutic potential of proteins, and how it translates into time and cost benefits. Sean, thanks for joining us.
Danny, it's great to be here.
We're going to talk about AbSci, its platform technology for therapeutic discovery of biologics, and its potential to improve the process of bringing biologics into development. But before we get into how the platform works, though, I thought we could start with the problem of discovery of biologics today. What problems were you trying to solve? What opportunities did you see for improving the process?
Yeah, so if you look at the biopharmaceutical drug discovery and biomanufacturing process, it's completely disjointed stepwise and has a couple key failure points that I attribute taking years to go from idea to drug in the clinic. And what we're doing here at AbSci is collapsing that into a single step that we call our integrated drug creation platform that allows us to go from idea to drug in the clinic in not years, but in weeks or months. And not only that, we're enabling new classes of biotherapeutics to reach patients that previously have been unattainable. Our partners like Merck and Astellas are so excited about our technology because they're able to take the ideas that they've always had on how to better engineer these protein-based therapies and actually make that a reality. And then finally, we're able to help lower the cost of goods as well by 50 to 75%. This is a truly holistic approach to drug discovery and development.
One of the consequences of this is you don't separate cell line development from discovery. Can you explain how that's normally done within the industry and what unifying this process does?
Yeah, so the two key problems that you normally have within drug discovery is first is the molecular reformatting. So you usually have to start off with phage display or yeast display, displaying an antibody fragment on the cell surface. And then once you find a hit or a candidate that has the affinity that you're looking for the drug candidate, you then have to reformat that into whatever modality you want to go into. For simplicity's sake, let's just say it's a full-length antibody. So you're going from an SCFE, reformatting that to a full-length antibody. And a lot of times what you see is the drug-like attributes you were screening for don't always translate once you've molecularly reformatted. So you have to then go and start the process over again or take a suboptimal hit forward. And the second key failure point is the transitioning between cell lines. So you have to start off with screening in phage display or yeast display. And then once you find a hit, you go into a transient cell line. And then from there, you have to build a stable CHO cell line. And a lot of times what ends up occurring is that pharma companies will find really exciting new hits, but by the time they find a CHO cell line that's stable, the yields and the quality isn't where it needs to be in order to progress it to the clinic. And so they ended up having to shelve these candidates. And what we've done here at AbSci is eliminated those two key failure points. Again, the molecular reformatting and the switching between cell lines. And we are able to discover the drug candidate at the exact same time we're developing the cell line. So we're doing simultaneous drug discovery and cell line development and collapsing this all into a single step. And one of the beautiful parts about this platform and technology is that we are able to screen billions of different cell and drug candidates all at the exact same time. So in a single experiment, we're able to look at drug functionality and manufacturability. So essentially looking at the drug affinity, the titer, and the quality. And that's what we're feeding into our deep learning models. But so that's a high level overview of how we're collapsing it into a single step and really changing the paradigm of drug discovery and cell line development.
You're using artificial intelligence in this process. As I understand it, you use this to make what you call non-obvious predictions about optimal drug candidate sequences and cell line design. How does your AI system work? What are the inputs it's considering, and on what kind of predictions is it making about the outputs? Is it giving you a gene sequence, an RNA sequence, a structure of a protein?
Yeah, so what we're feeding into our deep learning models to train them is data on the protein affinity, the titer, and the quality, as well as the target sequence that they're going to be binding to. And eventually where we're headed with all of this is once we've fed the models with enough data, we'll get to the point where we're able to predict the absolute best drug candidate for a particular target. So then the input would be the target sequence, and then the output would then be the protein sequence that has all of the drug-like attributes that we want, such as binding affinity, low immunogenicity, predicting the modality. And then additionally, again, this is a holistic approach. So we're also taking into account the manufacturing, being able to predict the associated cell line that then will manufacture at the high titers and high quality. And this is, really the future. It's our vision is to become the Google index search of protein-based drug discovery and biomanufacturing.
Well, let's walk through the discovery process with your platform. When most synthetic biologists talk, they talk about a 4-step design, build, test, and learn cycle. And I notice you have a process that sounds a lot like this, but has a few more steps in there. How do you go about target discovery?
Yeah, so we recently acquired a company, Totient, that had really exciting target and drug discovery technologies where they are able to take patient samples and be able to do bulk RNA seq on those patient samples and extract out new antibodies. And what we then do is pan those antibodies against all the proteins in the proteome to find new novel targets to go after. And so to give an example of this, Totient during the pandemic was able to take lung fluid samples from patients that had COVID and be able to extract out the antibodies from bulk RNA-seq data, and they found 15 neutralizing antibodies. And there's been papers that have been shown that there are different immune responses in different tissues and areas within the body, and so we're actually getting a very different immune response from the samples from the lung fluid than you would get from B cells and doing single cell sequencing from those. And so that then integrates into our platform to allow us to again go from the patient samples, discover the brand new targets, and then develop the best drug assets towards those new targets for all those various indications.
So how do you go from there to designing antibodies?
Yeah, so the great thing about the Totient platform is the antibody that helped you discover the new target can be the starting point for the drug discovery. So we can use that to determine if we should do further modifications to that antibody and put it in the modality that will work best for the biology that we're trying to solve for.
So from that point, you're able to construct a large number of genetically distinct versions of cells to make that protein of interest. How large a population of cells do you create, and how much variation is there in the end product? Are they different proteins or variations on the same protein?
Yeah, so we usually build a billion-member library looking at all the permutations in a CDR region, and we then screen that in our assays. And from there, we then down select to the top 1,000, and we then take those through more extensive screening, and also ensure that the hits that we have will also scale up for GMP manufacturing, so everything is fully scalable. And we utilize our deep learning technologies to help us predict the best billion-member libraries we should be using for that given target, and to be able to screen billions of different drug candidates in the exact format that goes into patients is a huge innovation, because currently being able to screen in the exact format that goes into patients, you're maybe screening thousands maybe or tens of thousands. We're screening billions, and so that's been a huge breakthrough. But if you actually compare that, though, to the variants in an antibody sequence, there are more sequence variants in an antibody than there are atoms in the universe. So when you compare atoms in the universe to billions doesn't sound like a huge innovation, and that's where deep learning comes in. Essentially, we're able to sift through all the potential evolutionary paths and predict the best billion-member library we should be using, or the best billion-member sequences we should be using to screen against a particular target or indication.
And once you have all these cells, how do you pick the best ones?
Yeah, so we the assay is built on flow cytometry, and so we essentially are able to through flow sorting pick out the top hits that have the high titer and the high affinity that we're looking for. And then from there, we take the top 1,000 hits and again, do more extensive characterization on those.
Are there bottlenecks in traditional cell development that your system is able to bypass?
Yeah, I would say one of the great things about our platform is that we are screening in an E. coli cell. So there's a lot of advantages screening an E. coli versus CHO or mammalian cells. First is cycle time. So we're able to move a lot quicker. We have 2-day fermentation times in E. coli versus 14 to 21 days in mammalian or CHO cells. Additionally, it's a lot easier to engineer and manipulate, and the ability to generate the billions of different data points and run the types of assays that we've developed. We wouldn't be able to do that with CHO cells. And the last piece is the ability to lower cost of goods by 50 to 75% by being in E. coli. And so those are the major advantages that we have when it comes to the cell line development.
One of the things you did this year is acquire Denovium, an AI deep learning company. You generate tons of data in your process. How have you incorporated the technology into your process, and what do you expect that payoff to be?
Yeah. So we are generating billions of different data points through our breakthrough assays, again, on protein functionality and manufacturability. And it got to the point where we're like, wow, we're generating so much data. Like, what better use than to feed all of this data into deep learning? And we always look at whether we should build out capabilities in-house or we should go out and acquire them. And we had an initial partnership with Denovium and had some great success, and decided that it would be best to acquire Denovium to bring in the deep learning capabilities in-house. And what we're currently doing is feeding in all of the data that we've curated and continue to curate into these deep learning models. And what it's allowing us to do is to predict the best billion member library we should be using for every given target that we work on. And eventually where we're headed with this, and what I'm most excited about is being able to go fully in silico. So as you train the model with more and more data, you're able to get more predictive. So instead of predicting a billion member library, you start predicting 1,000,000, 1,000. And then ultimately we're going to get to the point where we can take the target sequence, feed that into the model, and be able to then predict the absolute best drug candidate for that particular target, predicting the affinity, the modality, ensuring the lowest immunogenicity, what the half-life should be, the stability, and then also predicting the associated cell line to accompany it to ensure that you can get the high titer and the high quality. It literally all done at a click of a button. This is how we're becoming the Google of synthetic biology. Think of this as the Google index search of protein-based drug discovery and biomanufacturing. It's really the merging of AI and deep learning together, the merging of biology and deep learning together.
And have you had a proof of concept? Does it work? Can you point to cost or time savings or novel molecules that you've been able to discover that shows this is actually a better and more efficient way to discover drugs?
Yeah. So as you had brought up, we had just recently acquired them this year, and we've already had some really great breakthroughs and case studies that have come from the integration. And the first one is we were able to take a cell line development program we were working on and feed in that data into the deep learning model, and be able to we were able to predict and test a strain that had 2x the titer that we were able to develop on our own. And one of the chaperones that helped assist with the increase in titer was actually a chaperone that was not predicted to be a chaperone from basic bioinformatics tools. And so we actually discovered this new protein that we have found to act like a chaperone that was not a known chaperone, that increased our overall titers by 2x. And so that's just really scratching the surface of where this technology is headed. But in the short time that we've been integrating Denovium with AbSci, these are some of the exciting breakthroughs that we're seeing.
One of the other offerings you have is what you call bionic proteins. These have non-standard amino acids with chemical handles for modifications like pegylation and antibody drug conjugates. What's the range of things you can do with these, and how does it change the process of making things like an antibody drug conjugate?
Yeah, so the non-standard amino acids have been around for quite some time, but one of the issues with existing technologies is the ability to achieve high titers with the non-standard amino acids incorporated, as well as misincorporation or low incorporation rates of the non-standard amino acid. And we have solved both of these problems. We're able to get high incorporation rates along with high titers with the non-standard amino acid incorporated into the protein. And the exciting aspect of this is being able to have very site-specific bioconjugation. So wherever you want to put a bioconjugation, such as pegylation or an ADC warhead, you can use simple click chemistry to have a very homogeneous population versus having to use free cysteines, which can be a very nasty manufacturing process, and so it really opens up the ability to have very site-specific homogeneous bioconjugations, which increases overall efficacy.
What's the business model? Is the intent to only partner with drug makers, or are you developing any products on your own?
Great question. So our mission is to change the world one protein at a time, with the vision of getting the absolute best drug candidates to patients at truly unprecedented speeds. And with that, and with being a pure play technology company, we felt that for shareholders, as well as seeing that vision through, it was best to partner with large pharma and biotech companies where we develop the drug asset and the cell line, and then out-license it to our partner to then commercialize it and take it through the clinic. And we then are able to share in the upside. So we get milestones and royalties with the drug candidates and cell lines that we develop for our partner. So we get to share in the upside without having to take the binary risk of developing our assets ourselves. And it's a very different skill set as well, taking drugs through the clinic versus staying a pure play technology company. And so we wanted to really focus on what we do best and be able to serve as many patients as possible.
AbSci raised about $200 million through an IPO in July. How far is that money going to take you, and how's it being used?
Yeah, it is being used to really see our vision through. It's being used to continue to build out our overall capacity to recruit talent, and also being invested in ongoing R&D, being able to feed the data into our deep learning models to ultimately have us go fully in silico. So both on expanding current partnerships, our R&D, and continued talent recruitment and acquisition.
Sean McClain, founder and CEO of AbSci. Sean, thanks so much for your time today.
Yeah, thanks so much.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, Subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.